This overview highlights the core challenge of integrating diverse omics layers: genomics, transcriptomics, proteomics, metabolomics, and phenomics, where each dataset feels like a different puzzle piece with its own scale, noise, and missing parts. The illustration emphasizes how bridging those heterogeneous signals into coherent systems-level models requires careful alignment, interpretation, and thoughtful algorithm design. This slide shows why direct fusion often fails. Each omics layer has distinct statistical distributions, noise profiles, and measurement units, so naive concatenation produces mismatched data matrices. Practical solutions require layer-specific pre-processing such as normalization for genomics, imputation and filtering for proteomics, and transformation or batch correction for metabolomics before attempting integrative analyses that yield meaningful systems-level models. Here we focus on missingness and imputation bias, explaining that platform-specific dropouts in RNA-seq and undetected peptides in MS create non-random gaps which naive mean or zero imputation can distort. The recommended path is probabilistic gap-filling and uncertainty-aware methods that propagate confidence through downstream networks to avoid inflated correlations, false positives, and misleading biological interpretations. This diagram highlights how reagent lots, instrument drift, and sample processing days introduce systematic variation that can dwarf real biological signals, and it outlines harmonization tools like COMBAT, lemma, and quantile normalization. Layered scaling and careful batch effect correction are necessary to prevent a single high-feature dataset from dominating integrated analyses and to preserve true biology. The P much greater than N problem describes thousands of molecular features but few samples, which increases false discovery and overfitting risk. Effective mitigation combines targeted feature selection to reduce noise, factorization to reveal core structures, and regularized models to control complexity, producing more robust, interpretable models that tame overfitting while keeping biological relevance intact. This survey compares unsupervised integrators like MOFA, supervised approaches like Diablo, and network-based fusion methods such as SNF, pointing out that algorithm choice shapes the biological conclusions. The critical warning is that uncritical automation can flip results, so method selection must be guided by the specific question, validated with appropriate benchmarks, and interpreted in context. Missing metadata often obscures true molecular signals, as scant information on patient diet, sampling time, or cell passage can introduce latent covariates that masquerade as regulatory pathways. The clear call is for perspective, standardized metadata collection and annotation so models can separate technical or environmental confounders from genuine biology and become more interpretable and reliable. This cautionary slide explains how simple concatenation of omics layers can inflate pathway scores and create circular validation leading to false positives. The safer alternative is building sparse multi-layer networks combined with causal inference tests to distinguish driver events from passenger signals, thereby prioritizing validated therapeutic targets and avoiding premature, misleading claims. Single-cell and spatial omics add crucial cellular heterogeneity and positional context missing from bulk measurements, revealing individual profiles and tissue architecture, but they also multiply data volume and complexity. Resolving scalability and integration hurdles with advanced algorithms and computing resources will enable unprecedented mechanistic models linking genes, proteins, metabolites, and phenotypes at high resolution. Advanced computational approaches bring deep learning to cross-omics integration to capture nonlinear interactions, while highlighting current bottlenecks like feature selection, dimensionality, and noisy data. The slide also positions quantum computing as an experimental avenue to escape computational limits, emphasizing that any powerful method must be balanced with strategies to retain biological interpretability and robustness. The path forward rests less on isolated algorithms and more on shared, quality-controlled data ecosystems. Federated repositories for decentralized access, unified file formats for consistency, and ring trial reference datasets for benchmarking create the infrastructure needed for reproducible science and clinical translation, forming the foundation that lets robust integration methods deliver trustworthy results. These actionable steps lay out a roadmap, apply layer-aware pre-processing tailored to each omics type, capture and standardize rich metadata following FAIR principles, benchmark and validate integration algorithms against references, and build open data commons and collaborative infrastructure so the community can convert current frustrations into reliable, interpretable systems biology discovery engines.